This procedure starts with cDNA that has been synthesized from a template of high-quality RNA. The cDNA is first labeled with the fluorescent dye Cy3. Upon completion of the labeling reaction, the labeled cDNA is purified, isolated, and the quality of the labeling reaction assessed. The labeled cDNA is then hybridized onto a microarray. Following hybridization, the arrays are analyzed to determine relative levels of gene expression in the samples.
Hello, my name is Sam Peterson from the laboratory of Dr. Jennifer Freeman in the School of Health Sciences at Purdue University. Previously, we have shown you how to extract RNA from zebrafish embryos and turn that RNA into cDNA. Today, we'll show you a procedure for labeling and hybridizing that cDNA onto a Zebrafish Oligonucleotide Microarray platform to study global gene expression profiles. Typically, we use this procedure to study differential gene expression in response to an exposure to a toxin. So, let's get started.
The microarray experiment requires a minimal light environment to prevent photodegradation of the fluorescent dye Cy3 used in labeling. To start labeling the cDNA with Cy3, thaw the following components of the bioprime array CGH genomic labeling system: 2.5x random primer buffer, 10x DCTP mix, and nuclease-free water. In addition, thaw the 1 millimolar stock of the Cy3 labeled DCTP. After thawing, briefly centrifuge all reagents. In a 0.6 ml thick-walled tube, combine 500 nanograms of cDNA, 40 microliters 2.5x random primer buffer, and nuclease-free water to achieve a total volume of 86 microliters. Mix well.